So today we're going through development and its genetic basis. So practically, the story of how we get from like one egg and one sperm, right, the zygote of fertilization, how it produces a full functioning human by the end of pregnancy. But we're going to kind of like focus in more on that period in the first 8 weeks, right? Because in the first 8 weeks is when we complete early organogenesis. So by 8 weeks, we actually have the primitive structures of every organ system, which is pretty crazy. So the way we do that, right, the way that we get from one cell, one zygote, into a full functioning embryo in 8 weeks is through, right, differential gene expression, right? So what happens is we have two processes going on at once, right? We have a process for housekeeping genes and specialization genes. So housekeeping genes just means those genes that are responsible for processes that are required for the cell to just be alive, right? It needs to be able to repair. It needs to be able to synthesize DNA if it needs to. It needs to be able to proliferate if required, okay? So these housekeeping genes are always on, okay? No matter what happens, no matter whether you're in the heart, right? You're in the feet, you're in the head, it doesn't matter. The housekeeping genes remain constitutively active, right? The way that we get different organs in different places, and the way we get a functioning life that has a whole array of different functions across the body is by then changing how we specialize our genes, right? So these are always on because this is the essence of what it is to really be a cell. But the specialization is the process by which we make heart cells that can contract versus liver cells that can synthesize albumin, for example, okay? When we specialize, it's kind of important to specify that we lose flexibility, okay? Once you specialize your cell type, that's kind of the cell for life. We'll go into reversibility really quickly afterwards, but yeah, we say they lose flexibility as they differentiate. Okay. And the way that we specialize is we want to change the way a gene is expressed, right? We want to change the expression, like when it gets expressed as well as where, right? We don't want a contractile heart cell to be produced in the foot. We don't want, right, the liver cell that's synthesizing albumin, as we said, to be produced in the brain, right? They need to be turned on at the right time and at the right place. And the way we do that is through 4 main mechanisms, okay? First is gene rearrangement. This can happen on a chromosomal level, right? You can, during meiosis, you can cross over your chromosomes. Or it could happen at an individual gene level, right? And that's actually how you get like T-cell variability. You can regulate your gene expression through alternative promoters. What this means is just you're setting the start site of gene transcription at a different place. Alternative splicing, so that's like post-transcription. Once you're like cutting up your gene to form introns and exons, you can cut it in different places, right? And then non-coding RNAs, right? So what these do are these are RNAs that do not form a protein, but are functional. So they have a role in silencing genes, okay? They have 3 main mechanisms, right? They can cause promoter silencing, they can inhibit translation, and they can also stimulate the breakdown of mRNAs via double-stranded RNases. All right, and then of the types, right, you've got small ones. These are kind of mostly housekeeping genes. They can have a role as well in like structural functions, so structure and function, so like the structure of a chromosome. Large non-coding RNAs may be involved in like imprinting, which we'll go into very briefly later, X inactivation, things like that. And then your small interfering RNAs and your microRNAs. MicroRNAs are involved in gene regulation. They actually have an important role in disease as well. Like if you have a misexpression of this, you're not able to silence a gene, like an oncogene, and you might get cancer. Small interfering RNases, you can get exogenous or endogenous, right? Endogenous being retrotransposons, exogenous could be viral or man-made, and they're used in treatment as well. I said we'd come really quickly to reversibility. So I said that as we change the expression of our gene, we specialize, right, and we lose flexibility. However, it is actually possible to reverse the process. So once you've produced that heart cell that I keep coming back to, you can actually reprogram it, right? Well, this is the future, right? We're trying to be able to reprogram it to be back to that totipotent zygotic cell, right? Where you can produce any cell in the body. And the way we do this is with reprogramming factors. And at the moment, we have viral reprogramming factors and, what's the other one? Viral and, the other one slips my mind, but transposon, there we go. Viral and transposon-based reprogramming factors. And yeah, so what you can do is the current research is if you take like an epidermal cell, you can give them either of these programming factors and kind of turn it back into that embryonic totipotent state, which is pretty cool. Yeah, and you can like apply that for anything. If you have heart disease, hopefully you can regrow a heart. You can regrow substantia nigra if you have Parkinson's. There's a whole host of applications. In terms of genetic basis of development, we also have what's called the human organizer. So these are cells of the primitive node. So if you think to like early embryonic development, you have your primitive groove, and then on either side you have the primitive node. And these primitive node cells, they have specialized cilia, right? And these cilia have specialized angle and structure such that when they beat, they cause like a leftward beating current, okay? And what this does is then it moves calcium ions to the left more than to the right, okay? And then you get a buildup of intracellular calcium on the left. And what this does is it initiates a genetic cascade that is different to that on the right, okay? And this is how you get body asymmetry, right? Not only facially, you may have facial asymmetry, but also like internally. Like your heart is on the left, the liver is bigger on the right? This is all to do with the human organizer. And just briefly talking about epigenetics, so epi, above, genetics, genes. The way we can use genes to like alter development is through inheritance of epigenetic marks. So epigenetics doesn't necessarily have to always be acquired. Doesn't always have to be inherited. It can be acquired. But in this case, we'll just talk about genetics, inherited epigenetic marks. So we have three mechanisms to alter a gene. And to clarify, when I say altering a gene in terms of epigenetics, it means changing how it's expressed, but not the sequence itself, right? So the three mechanisms, you can like methylate your DNA at your cytosine residues, happen at your CG groups, also known as your CPG islands. And these often occur near to promoter regions. So when you methylate, you inhibit the binding transcription factors and influence the expression of the gene. The second mechanism is kind of similar, but for histones, there's like a tail on a histone protein. And if you methylate that or acetylate that or ubiquitinate that or citronate that or whatever you do, right, it influences how tightly packed the DNA can wind around the histone. And the third mechanism is actually the same as these, the non-coding RNAs, the silencing that we spoke about. And so genomic imprinting is a process by which we can inherit these methylation changes from a parent, okay? And these, a quick note, they override the laws of dominance. So if you have if you're heterozygous for a particular trait, so you have one copy of the dominant allele and one of the recessive, and you have inherited a methylated dominant trait, you're actually going to express the recessive trait. It doesn't matter that you have the dominant trait, and Mendelian genetics would tell you you would express it. Epigenetics kind of overrides that, it trumps that. There's just a quick example here of how epigenetics might affect development. So the IGF-2 H19 locus. Just really quickly how it works is IGF-2 promotes growth of a fetus, H19 causes growth restriction. And that's why in a father they have high expression of IGF-2 because they want to promote the survival of the fetus, whereas from the mother there's high H19, which is higher growth restriction, which might seem counterintuitive, but they need to ensure that the fetus doesn't grow too large that it compromises the womb and no future offspring can be produced. And so where epigenetics comes into all of this, with the mother, the reason they have higher H19 H19 expression is because they have decreased methylation of the imprinting control region, ICR. And what I've written here is CTCF first methylation. So with maternal genes, the imprinting control region is not very methylated. So this CTCF protein can bind. And what it does is it comes to inhibit this connection here, where the enhancer is stimulating IGF-2. So if CTCF can bind because of the lack of methylation, there's more inhibition, reduction in IGF-2 expression. Therefore, we have a higher ratio of H19 to IGF-2. Whereas the opposite is true for the father. If you have higher methylation, right, less CTCF can bind, disinhibition, greater IGF-2, and you're going to have greater fetal growth. And just some final remark on some causes of epigenetic changes. So they can be environmental. So mothers who have been starved, for example, have decreased methylation. The higher the folate intake of the mother, the less methylation occurs, and it actually changes this IGF-2 H19 locus. Things like benzene rings can cause epigenetic marks that cause blood cancer, air pollutants, pesticides, all things like this can cause these epigenetic changes. And as kind of like one small piece of the puzzle, when you put all of this together, you kind of have the genetic basis of development.